So you pair this protein with an mRNA, or not mRNA, a gRNA, and you pair these together and it goes to a specific spot of a human's DNA, and then it snips the DNA and then you can recover it in some way. So that has been conceptually really amazing over the past like decade or so, but hasn't really been used on people yet. It's still mostly in trial stages of like, in theory, we can change people's genetics to do various things, but we don't. This is very difficult in practice. So then a few weeks ago, there was a baby who was born with CPS1, which is this basically extremely serious genetic disorder.
Okay.
Untreated, you've got like a week and you die.
Jesus.
And treated like aggressively, like the baby's living in the hospital for months or years, they're maybe gonna survive with severe neurological damage. So this is like an incredibly severe disease that is caused by a genetic mutation, like one gene that is basically wrong. It's like hundreds of people get this a year. It's not massive, but this baby was born with it, I think about 9 months ago. And immediately upon being born, they recognized that it's this horrible genetic disease. And this team set out to try to solve it using CRISPR. And so instead of the normal like decade to develop something custom, they did this over 6 months. And these doctors said like such speed to producing clinic grade CRISPR for a genetic disease has no precedent in our field, not even close. These steps traditionally take the better part of a decade, if not longer. But they managed to essentially map the exact genome of this baby to map exactly what their DNA sequence looks like, find using computer modeling the exact spot that was mutated in the genetic sequence, design a molecule through gRNA that can find that specific spot and then cut it and cause the body to basically replace it with healthy stuff. And it worked. So they saved this baby's life a few weeks ago in May. This is the first time ever that CRISPR has been personalized and used on an actual baby. So they were able to like modify this thing, a genetic treatment for a specific person. And they're saying now that it's done, future treatments could be a lot cheaper by an order of magnitude at least, is what one of the doctors said. So CRISPR is maybe you've heard of it if you followed biology at all. Incredible potential to this thing. Like, for example, in theory, we can cure Huntington's disease, which kills like 250,000 people. I don't know if that's a year or in general who have it, but that's fatal. That's a single gene mutation. We could like cut and cure that. Alzheimer's risks. You could find the genes that increase the risk for Alzheimer's, edit those, which I think many, tens of millions of people every year who are dealing with that. Curing HIV. HIV is crazy because it gets into your DNA, like it embeds itself into your DNA, and we could, in theory, have CRISPR that goes in and like snips it. And then even the changing people's genes before they're born is where it gets kind of crazy sci-fi. And there's a famous example of this in 2018 where this scientist in China just went solo and just edited embryos. I don't know if you guys heard about this.
That's scary to me.
No, this is still good news. So he talked to patients. He like found some people, like couples who like one parent had HIV and the other didn't. So the baby's at risk for HIV. And then he edited embryos and then implanted them into the women. And one of the women had twins. And he went to jail for 3 years because the entire scientific community was like, what the fuck, man?
I remember this.
Yes. It's like,
yeah,
deeply unethical shit. So, but the wild thing is it could work, And so while there's many ethical questions right now, and CRISPR isn't perfectly precise, there's various things. One of the things to remember is that you have a copy of your DNA in every cell. So it's not like you go and change one cell. You sort of have to get the changes to propagate through the whole body. So there's challenges with that and making sure it hits the right thing.